Good afternoon, everyone, and welcome to the BPN-Biologics Q&A webinar. My name is Mario Skiadopoulos, and I'm the Program Director for BPN-Biologics. As you may have just heard the automated message say, this webinar is being recorded. We will make this publicly available after this is completed on our website for your future reference. We will also make a PDF copy of slides available to all registered participants. If you have any questions, please post them in the chat and they'll be compiled at the end of the presentation, and we'll take a little time to go through those questions. If there are any questions that you may want to ask after the presentation, we'll be providing additional information where you can send inquiries to. So BPN-Biologics is a relatively new program at the National Institute of Neurological Disorders and Stroke. It's an extension of the BPN small molecules program that has been up and running for a number of years, quite successfully. It's expanded to support a variety of biological modalities and disease indications and entry points. I'm not going to go through all of the slides that I have in this presentation. Most of these here can be used for reference in the future if you'd like to go back and see a specific area that you might want a little more information. But I want to touch on some salient points on most of the slides. So I'll spend more time on some rather than some of the others. The goals of this program are to provide non-dilutive grant funding and all the necessary resources, consultants, contract services as required that are typically lacking in our research community. We'd like to de-risk potential therapeutics to the point where industry will be interested in investing in them. And this typically happens after a phase 1 trial where safety and has been shown and efficacy has been demonstrated in animals. Therapeutic candidates become much more interesting to venture capitalists as well as industry groups. Importantly, we preserve the institution's intellectual property. We don't have any stake in that at all. This program can provide support for both academics and small businesses. This is through 2 different opportunities for funding, 2 different FOAs listed here, 163 for academic groups and 233 for small businesses. And we'll support biotherapeutics discovery through a phase 1 clinical trial, depending on at what point the program starts and how much work is actually done within the 5 years that are allotted. And I'll be talking a little bit more about this. The next receipt date is August 9th. It's coming up very soon. BPN-Biologics, just like BPN small molecules, is a multi-IC, multi-institutes and center program. We have here listed a number of the ICs that institutes that are and centers that are supporting this program. Depending on your particular area, if your grant is successful, if your application is successful, it'll wind up going to one of these institutes. But everything initially comes to the NINDS and is reviewed in a single review by one team. It's not different teams for different institutes. Here are listed the main mission statements for each of the different ICs. So really depending on where your particular disease area is, it may fall into one or another IC. Sometimes there's some overlap. So we discuss that, and we have some ground rules, if you will, as to where a particular project may fall into. So what does the BPN-Biologics Network look like? We've got an external oversight committee shown here at the top of 5 to 7 members that provide high-level guidance for the program. They meet 3-4 times a year and provide their opinion as to where we should be looking at for improvements and how the program is doing overall. But the center of the core of each of these awards is the lead development team, which is comprised of the principal investigator, the investigators team, and industry consultants that the NIH provides, who are experts in a variety of areas, and I'll discuss that in a little more detail briefly, as well as NIH staff, including program managers, project managers, and subject matter experts in various neurological disease areas. So the FOA is written broadly to account for a variety of modalities. These can include monoclonal antibodies, nanobodies, peptides, nucleic acid therapeutics, viral vector therapeutics, protein-based or protein subunit-based therapeutics. Anything that's typically considered a biologic would fall into this program. If it's not a biologic and it's a small molecule, as I mentioned earlier, there is another program that also has a receipt date coming up in August 9th of this year that can handle small molecules. And it's similarly structured in terms of support as BPN-Biologics is. A little bit about the contract resources we provide. They can be lumped into 2 categories. We have the preclinical services, which includes manufacturing, CMO services for pretty much any biologic that would be required for developing a therapeutic for a neurological disorder. That includes, as I mentioned, ASOs, peptides, cell-based therapeutics, and these groups will conduct process development, analytical development, small scale, large scale, cGMP manufacturing, formulation, and fill finish. Everything required to basically get to the clinic. And we'll also be providing preclinical services, which include animal studies to demonstrate safety, pharmacokinetics, pharmacodynamics, bioassay validation as needed for the clinic, as well as for animal safety studies, and the serological testing for both clinic and pre-IND IND-enabling studies. The second set of contracts that we have is the consultant subject matter experts. And this is a team that is an international team, really, US as well as Europe and Australia, experts that are seasoned industry expert in the areas that are indicated here, including CMC for a variety of biological modalities, regulatory affairs, non-clinical studies such as I mentioned pharmacokinetics, PK, PD, ADME, toxicology studies. We have medical writers, and we'll have quality consultants that can help us with any quality issues or concerns to make sure that we're doing everything the way we should be, both internally as well as at the CROs and CMOs that we're contracting. And of course, we have biostatistical support to ensure that studies are properly designed and meet the rigor requirements of the NIH. So how is the program structured? Generally speaking, it's divided into two phases, the discovery phase and the development phase. And depending on whether you come in as under an academic application or a small business application, there's different terminology for these two separate phases. For SBIR, it's called phase 1 and phase 2. For the academic application grant, it's UG3, UH3, UG3 being the first phase, the discovery phase. And there's also very important to note time limits on these, 2 years at the most for the discovery phase, which is really lead optimization, and then up to 4 years for the UH3 or phase 2. And the number of years in this phase really depends on how many years you've used up in your discovery phase. So you can go up to 4 years, but typically these would be 2 and 3 years. And during the development stage, you'll be conducting manufacturing of the lead candidate, CGMP, clinical lot material, as well as material that can be used in IND-enabling toxicology studies and PK studies and any other studies the FDA may have requested be conducted with that type of material. In the announcements for both of these funding opportunities, I encourage you to go to the website where these are posted and read them from top to bottom very carefully, because we have to go by what is written in those FOAs. As I mentioned, up to 5 years, no more than 5 years for each of these mechanisms. There are funding caps that must be adhered to. For the UG3, UH3, it's not technically it's not limited, but must reflect the actual needs of the project. For U44, it could be 500,000 a year for the first 2 years, and then 1,500,000 per year for the next 3. But it's really important, no matter what your budget is or what you think it looks like, to run it by the program director for either your institute or you can contact us. It's good for us to take a look at the budget and make sure that we don't need to get special approval. If you are over the caps, the limits, we need to get approval, and it takes time for us to review and to present to the committee that reviews budgets that are above the limits. So you need to contact us. I would say at least really 8 weeks, or even give us more lead time if possible, before the deadline for the application, which this year is August 9th. Foreign organizations can also apply. It's generally speaking better if they are collaborating with a U.S. partner, but that is not a requirement. If the primary institute is a U.S. organization, it does make it simpler. That being said, as we've stated here, that if there are special opportunities, resources available outside of the U.S., and these are resources or candidates, therapeutic candidates, for example, that really don't exist or not being developed in the US, we will certainly consider these applications as well. Intellectual property, another very important consideration. Please, again, read the FOA carefully. The NIH does not have any stake in IP that the PI or the PI's institute has, but they need to have some kind of IP in order for this to move forward. So, and that needs to be described in the application. A little bit about the grant mechanism. So the 5 years are divided up into a number of milestones that need to be completed each year. And then there are go and no-go decision points after each milestone and after each year. So the transition is not like it is for many other grant mechanisms. There are a number of points where if a milestone has not been met and it seems to really be a no-go point, the grant will be terminated at that point. At the end of the second year, I don't know if I have can't remember if I have a slide for this coming right up, but at the end of the second year, the lead candidate needs to be ready for manufacture. So that means the first thing you're doing in year 3 is gearing up for putting that IND package together, which includes manufacturing material for the clinic, manufacturing material for your IND enabling studies, and executing those IND enabling studies. So there's really no other activities, no further optimization after year 2. So keep that in mind as you're planning your activities. So entry criteria for the earlier stage, the discovery stage. You need to have identified 1 or more leads that are sufficiently profiled so that the parameters that still need to be optimized can be quantitatively specified. And these will become milestones, specific aims, of course. If you're looking to optimize a specific characteristic, you need to have clearly defined and quantitatively specified optimization expectations. You need to come in with very good, solid preliminary in vivo efficacy data in an appropriate animal model. Assays that are required to support the program need to have already been developed. So the program itself is not really supporting developing assays. These should have been developed and come into the program ready to use. They can be qualified and validated for use in the clinic and in IND enabling studies. And of course, there's, I just mentioned, no IP constraint. The end goal after the discovery stage is to have a lead clinical candidate that has all of the target characteristics that were stated in the original application in terms of manufacturability, stability, efficacy, bioavailability, ready for manufacture at the end of the 2 years. You can also enter the discovery stage if the program is your program is currently at that point, and you've already gotten a lead that's appropriately optimized and ready to go. So the types of things you'd be doing at the development stage is, as I mentioned, manufacturing and conducting the studies required to satisfy the FDA that you're ready to get into the clinic. And that's This slide's basically what I just said. The minimum requirement for these applications is that at the end of the 5 years, you have submitted an IND package to the FDA. That is an absolute requirement. If you are able to, you can also begin a clinical trial. So I've got that here for your information, and depending on what stage you enter and how much work needs to be done at each stage, you can actually initiate a clinical trial. If this is your intention, we advise you to reach out to us and discuss your plans with us so that we can provide guidance. General tips for applications. Please read the entire funding opportunity announcement carefully. There's a lot of information there and requirements and expectations that I've seen are very often ignored, and they will not be ignored during review. And we do a preliminary review, and if we find some box has not been checked or something that the FOA does not permit, we will have to contact you and ask you to, if it cannot be remedied, to withdraw the application. So very important to read the announcement carefully to make sure that your program really lines up with the expectations and that it is at the right stage for BPN-Biologics. Take the time to talk to program director for your home institute's disease area, just to get feedback. That's always helpful. If it's an SBIR grant, you can contact the SBIR PD for any issues regarding small businesses. You should really have solid scientific preliminary data to support the proposal. We're looking for projects here that really are ready for prime time. They may need a little bit more tweaking, and that's done in 1 or 2 years. And you really can't get that much done in 2 years if your candidate is not really ready for that stage. Okay, so it's very important to be certain that you really do have something that's ready to go, and that the data that you're providing supports that notion, because the reviewers will be looking at that very carefully to make sure that the current lead or leads really are ready for subsequent development and manufacturing. Clearly indicate in the grant what work you will be doing and what work you expect the NIH contracts to be conducting. We've seen a few examples where applications came in and they had sections of exactly what they wanted to do, and not really any information or very little information regarding the studies that they would like the contract services that we provide to execute. The way I would suggest you look at it is describe all the studies you think are required to get to an IND submission, regardless of who is doing the study. Describe them in sufficient detail so that the reviewers understand the purpose of the study, the intent, the expected outcome, and you can simply just mark in a table or any other way you think is acceptable, internally executed either through your own facility or through one of your contractors, or whether you want the NIH to contract to use our contract services. If you're using our contract services, you do not have to budget for that. We will pay for that. So just note that it's not included in your budget because you're expecting the NIH to pay for those services. Everything else, of course, needs to be budgeted. And include the budget page for all of the years of the grant. This is very important, 1 through 5. And don't shortchange yourself. If you think you might be able to get something done sooner rather than later, In reality, the real world, these things wind up taking longer than you think. So give yourself sufficient time to complete all of the activities required. Rigor is a very important consideration and will be when this is being reviewed by our panel of experts. It's important to explicitly discuss the quality of the data that you've presented, especially if it's in prior publications. You need to basically critique the data that you are presenting in those publications, address whether it was done in a rigorous manner, randomized, blinded, what power, all of that will be looked at, and it's important to make sure that show that you are aware of that, and if there's shortcomings, point them out. If there are strengths, of course, point those out as well. Regarding your approach for the studies that you need to execute, you should, as indicated here, provide details for controls, include details of experimental design. So it's important to get all that data, all that information available to the reviewers for them to be able to evaluate whether these are appropriately designed. I wanted to spend a couple of minutes on this section. This is specific aims. This is one of the first things we look at typically when you reach out for feedback about your program. We ask for you to put together a list of specific aims and a budget so we can get a sense of whether the aims are really in line with what BPN-Biologics supports. I've put this up here as an example. This is not for any one particular project. This could be for really any modality. Some of these will not be required for some modalities, and there'll be other activities here, not included here, that one may need to do. But the types of things that you should be thinking about putting in your discovery stage versus your development stage are listed here. Of course, on the left-hand panel, lead optimization. This is the area where you are the expert in, and you should really spend some time here describing what you're trying to achieve. How will you know that you have achieved optimization? What does success look like? What does a no-go look like? Manufacturing, small scale for the material you'll be using in these 2 years, whether you do it in-house, contract it yourself, or whether you would like for NIH to generate any material. These should all be listed out. Think about delivery routes, dose range, bioassays. This is something that's really very often ignored or started too late in many projects. You will require bioassays to measure the effect of your biologics, your biologic, the PK of your biologic. If it's expressing, for example, another gene product or suppressing a gene product, you want to be able to measure that, not just in your efficacy studies, but you'll need to have assays that are qualified or validated, depending on what the FDA expects, for your IND enabling studies, such as your toxicology study, your formal GLP PK study. These assays need to be ready to go. If you don't start getting those in place in the first 2 years of your program, they won't be ready to go by year 3 or 4. And of course, when you get into the clinic, you'll also require similar assays to measure activities or the product itself. And those need to also be qualified and or validated, depending on discussions with the FDA. That needs to be started early. So don't ignore bioassays. And of course, these will be different for different products, different modalities. The only other thing, most of these are self-explanatory. The last thing in your specific aims for year 2 really is that you've selected a lead candidate that has certain specific attributes that you've been targeting, and that's how you know that you're ready to go to the next phase. Another important consideration is an FDA meeting, either a pre-IND meeting or a pre-pre, now called an interact meeting, where it's more informational with the FDA. You really should get one of these done before we move to phase 2 and start manufacturing and doing very expensive manufacturing runs and IND enabling studies, which could run in the multi-millions of dollars. You really want the FDA to tell you that you're on the right track or not, that you need to let them tell you what else you may need to do, or how you may need to consider doing something during your manufacturing or toxicology studies. That's not something to do after you've completed your studies. So this really should be built in at the appropriate time point. And we will provide regulatory support when the time comes. We'll have regulatory expert consultants to help with that. But all of these really should be taken into account when you're thinking about your specific aims for the first 2 years. The specific aims for phase 2, and again, all of these will vary depending on your own program, your own needs, your starting point. But they'll probably be divided into 2 general categories: manufacturing, process and analytical development has to be done, small scale, possibly engineering runs, and finally, clinical scale cGMP manufacturing. And then all of the activities required for releasing this material, fill, finish, release, and of course, stability. Your stability, keep in mind, you need to demonstrate stability of your product through your clinical trial. And so if you've manufactured in year 4, for example, and your clinical trial is in 18 months, you need to know that it's going to be still potent and stable and efficacious 18 months after the manufacturing date. So you need to have accumulated sufficient stability data during these 3 to 5 years to show that the material you put into the clinic and into your IND enabling studies is still good material. And of course, all the pre-IND enabling studies, sometimes they need to be conducted with the actual clinical material that's actually going into humans. Sometimes the FDA can accept an engineering run that's been manufactured in a similar manner. It really depends on their requirements and what we're able to do within the time frame. But ultimately, the FDA has the last word on what's okay or not okay. The absolute requirement, as I mentioned earlier, is the IND package preparation and submission. This has to be part of your proposal. If you think you're able to, you can initiate a clinical trial. And by that, I mean, if you feel that there's sufficient time remaining, you can actually start a trial. If you're going to start a trial, you need to have been preparing for that, and that usually takes some time. So these are just examples of the types of specific aims you should be thinking about, including in your application. These will ultimately become milestones for a successful application that's been awarded. We'll take these specific aims and working together with our internal experts and consultants, we'll change these into milestones with go, no go decision criteria and success criteria at every stage. Another thing that I find very helpful, I think this is really helpful for reviewers as well, is to put all your activities down on a figure such as this. This allows you to do a number of things. First of all, you and reviewers can see where all the different buckets of activities fall into and approximately how long it's taking. It allows you to see if you've given yourself, have you really given yourself enough time to do something. For example, you can't do your toxicology study until you've made material acceptable for a GLP tox study. So depending on how long this takes, you might be able to get away with an engineering run that's representative of the clinical grade material. But you want to be able to demonstrate that all of these activities can actually fit into the time that's allotted. And then remember, there's a hard stop here at year 2. Year 2, you need to have a final candidate selected that's ready to go into the next phase. So it's a pretty tight, but it can be done. Okay, so these are not requirements, but I think it's very helpful for anyone looking at your proposal to get the big picture in one shot. Okay, a few things you need to be thinking about doing before August 9th. And these include completing your registration well in advance of the receipt dates. As I mentioned earlier, if you have a budget that you think might be exceeding our limits, please contact us. We need to discuss the budget, and then we need to have a review internally, which takes a number of weeks. So this is not something we can do in July, for example. It's going to be too late. It needs to be done as early as possible. And of course, you want to make sure that you've got all of your IP and licensing in place before you submit your application. Things not to do. Don't plan for this to be the sole funding for your laboratory, especially if you're thinking about providing support for graduate students or postdocs. These types of programs are high risk. A milestone, if it's not met, could mean that the program may end abruptly, and that means funding is cut off. So please keep that in mind. Don't under-resource your budget to avoid the limits, because that's not going to solve the problem. If you require twice as much as you've actually put in there, you're not going to be able to complete the studies anyways, and then you don't meet your milestones, and the program gets terminated. So it's really important to make sure that you're doing the appropriate studies with the appropriate budget. If there's some study that can be contracted through NIH, you can do that. We can talk about whether our contract facilities are able to execute those studies, and those studies will not be part of your total budget. BPN Biologics does not contract for disease biology or one-off experiments. Keep that in mind. How you designate your application is very important. If you don't code your application as a clinical trial if you plan to use the NIH clinical trial facilities. Okay, all this information is available online, but it's important. Otherwise, that could present problems. If you do plan to run your own trial, it should be designated as a delayed onset trial. Again, these are defined on the NIH websites. For more information, please take a look at our BPN-Biologics website. We have a number of pages here that can provide additional information that I may not have gotten into today. I also would like to remind you that if your program really is not a biologics therapeutic, or if you have a question of whether is it a biologic or is it a small molecule, there is another funding opportunity. This program is run by Dr. Charles Sywin. He's the program director for BPN small molecules. And this is another opportunity for developing similar type products that are small molecules, but getting to that same endpoint of IND submission and clinical trial. So that ends the presentation portion. We have a couple of Q codes here for you to look at. For one of these is our website. We have an FAQ on that website that you can access through this Q code. We'll be updating that over the next week or two, but it's available there for you to look at now, and just keep in mind to look at it again in a couple of weeks for any updates. If you have any questions over the next couple of months before the applications are due, please send them to this email here, BPN-Biologics questions, along with your specific aims and your budget. There is a link on our primary web page that has a PDF questionnaire. If you fill that questionnaire out, it provides space for your specific aims and a number of other questions that will help us in getting you the answers that you need. Send that PDF to us, and once we look at it, we can either respond to you by email, or we can set up a meeting and we can talk a little bit more at length about your project. Send your questions to this email. This will go to myself and to Elena Barnaeva and Shruti Thomas, who are health program specialists supporting this program. I might miss it if you send it directly to me, or if you send it to someone else, it might be missed. But if you send it here, we all get it. So there's a much better chance that we'll be able to immediately respond to you. So please bear that in mind. And that's the end. I think now we can do questions and answers. Elena?
We can start with a question that was submitted through email, and while everyone fill in their questions in the chat. The question is, will UG3, UH3, and U44 applications be reviewed together by the same study section and have the same impact score threshold for funding?
The answer to that is yes. All of these, regardless of which FOA they're responding to, are reviewed in the same study section. There is a benefit to the SBIR. It has a different bucket of funds available. So just that's another consideration. It gives you basically a better chance if you're a small business, because you don't have as many applications competing. But that aside, the cutoff scores, of course, they're similar. They will be similar from year to year. It really depends on how they're ranked in the end. So there's those criteria don't change. It's not necessarily easier via one mechanism versus another, but for budget considerations, there's different buckets of funding in one versus the other.
The next question is, do you support brain tumor study?
Brain tumor, I think that would fall into NCI. We'll have to get back, but I'm 99% sure that that's not. So what kind of tumor? Are we talking oncogenic tumor? I mean, it sounds like a cancer project rather than.
Well, if it's a biologic, if it's a biologic application, it might be probably considered. Right?
No, not if it's a tumor. But we can get back to you.
Mm-hmm.
Yeah, I'm pretty sure that would have to go to NCI.
The next question is NINDS specific. Are these cross-funding opportunities between BPN-Biologics and HEAL UG3 UH3 programs for pain therapeutics?
I'm not sure I understand what the cross-funding refers to. HEAL is similarly structured. It has a development stage and discovery and development stages and targets getting into the clinic early. Maybe we can unmute Elena and just have the person pose that question. Give me a little bit more background.
Yes, Yuri, please unmute yourself if you want to clarify the question.
Hi, this is Yuri Miller. Basically, my question is, can I submit a proposal to BNP-Biologic on pain therapeutic?
Oh, yes. Yes, absolutely. Yes.
And if
so this is for HEAL, okay, not for BPN-Biologics. For if it's a pain therapeutic, you can submit to HEAL, and they take both small molecules and biologics.
so it means that I should not submit a pain therapeutic proposal in response to this particular BNP-Biologic RFA, correct?
That's correct. Yeah. So there's that would fall within the HEAL purview. And that's Marianne Pellimonter. We can provide you with her contact information if you need that.
Thank you.
Thank you, Yuri. So the next question is, do you have an established support pathway for cross-purposing of molecules? And B, given a target creating viable small molecules from established large molecules?
I'm not sure I understand the. Can you repeat that first part?
Do you have an established support pathway for cross-purposing of molecules?
Can you maybe provide a little more information? What you mean by cross-purposing?
It's hi, it's Jan Johansson here. So, for example, we have a class of molecules that seem to work for Alzheimer's disease, but the actual target has viability for other indications. For example, eye indications, certain eye indications. So, in this instance, we have a set of molecules, and most of the data is based on CNS, but the point would be to cross-purpose this class of molecules for an eye indication. So what I just described should be a relatively common issue. And my question is whether you have addressed such issues before and have thought about how to establish support for that.
So if you've got something that you know works for one indication and you'd like to see if it works for the other indication, it's probably not ready for BPN-Biologics, but there are other funding opportunities where you could examine its potential efficacy. The reason for that is in order to get into BPN-Biologics, you need to have already demonstrated activity for the intended indication.
In vivo activity.
Correct.
And target
Correct.
engagement. Yep.
Yeah,
Yep.
correct. But
Yep.
if you were to go through, for example, Ignite or one of the other funding opportunities, there's also, I don't know if you're a small business, there's the SBIR Omnibus. You can get that data, you can generate that data, you can show, for example, that it actually is quite effective for another indication. Once you've obtained that data, now you're in a position where you can apply for BPN biologics because you have that prerequisite data already generated.
Okay. Okay, my other question there, question B, is probably more complicated, but let's try it. So, it's said in drug development that as you learn more about the novel target, you perhaps have the easy shots, the low-hanging fruits, by creating large molecules of various types, peptides, antibodies. But then eventually, as you learn more, it may be possible to create small molecules, which of course have their convenience upside, convenience and compliance for eventual treatment and an indication. So do you have expertise with experience in sort of transitioning established large molecules to small molecules for given targets?
We don't have that expertise in BPN biologics per se. I don't know if the institute does or not. I mean, what you're saying is you're probably going to require computer modeling where you understand, for example, how the biologic interacts with its target, and you can substitute the biologic with a small molecule that does the same thing. That sounds to me, it's a lot earlier stage activity. We can certainly provide funding for that. I really don't know. I don't think we have expertise in that per se. That seems to me, unless I'm misunderstanding what you're saying, that it's really heavily based on computer modeling to do something like that. But there are funding mechanisms whereby you'd be able to pursue those types of studies. Those are much earlier stage than BPN biologics.
Thank you.
The next question is, could you share previous application review experience for this funding mechanism? If there is any?
I can. I try to during the presentation. For example, make sure you've got all of the activities you believe are required, whether you need to conduct them in your own lab or through your own contractors or through NIH contractors. You need to describe everything from the year 1 through year 5. We saw applications in the past that did not have all of the activities listed in sufficient detail. They need to be really detailed, okay? Doesn't matter who's going to do the activity, whether it's a tox study or manufacturing run, they really should be described in sufficient detail so that when the reviewer is reading the application, they really have a good sense that what you're proposing makes sense and that it would actually move the program forward. That was one area where we saw some deficiencies. I'm trying to think where else. Sufficient preliminary proof of concept data, very important. That's directly relevant to the candidate you're proposing to bring into the program for optimization. Make sure that you've really generated that data, and sometimes you need another year really of data generation before you're ready for BPN. And the reviewers will see this. If you're trying to generate additional data while optimizing and it's not quite there, you're not going to be able to squeeze it in the 2 years that you have allotted. Really the way to look at it is that you on the day 1 of year 3, you need to have your candidate in hand ready to send to the manufacturer. Okay, so anything you propose before that really needs to get done in 2 years. There's no extension allowed. It has to get done in 2 years. So if you list out all of your activities and you see that you're at 3 years to get to that stage, then you probably should consider another funding mechanism that we may have available, such as create Ignite, that will give you a year or two to do a little further work on your candidate or candidates, so that it's in the right position for entering BPN Biologics. I'm trying to think of what else. Do you remember anything else, Elena, that we saw in that first round? The most common really was not including an IND submission and all of the activities required to support that submission. It's all got to be in there, specific aims and details. That's really, I think, one of the most critical things.
Yes, even if you are not planning to do these activities yourselves, you have to describe them. That's what they're looking for. They still need to have the idea what your plan is, even if it's done by the contractors. But I think it was the main concern of the reviewers, that people do not submit the plan for contractors' activity. Should I read the next question?
Yes, please.
How about cell therapy for pediatric disease such as perinatal asphyxia? Is it responsive to this program?
Cell-based therapies are.
Yes.
If I'm not familiar with that disorder, if that is a neurologically based disease, then the answer would be yes. The requirement is that it's a neurological disease or disorder. And if it's a biologic, such as cell-based therapy, yes, we can do that.
The next question, do I correctly understand that this program covers more advanced study, such as moving to production, but not as proof of concept?
That's correct. If you have not yet generated proof of concept data for your candidate, then it's not really ready for BPN Biologics. You need to have generated proof of concept data, show that it's targeting where it's supposed to target, it's doing what it's supposed to be doing, it works. You might need to tweak it a little bit, optimize it, work on the PK or affinity, for example, but you've already generated the data in the appropriate animal model that will convince reviewers that this is ready to go to prime time. And if it's not ready for biologics, again, there's other funding mechanisms that could give you a year or 2 or 3 of support to get to that stage.
Is a comparison of AAV serotypes eligible for lead optimization? This will be followed by route and titer optimization.
Yeah, that's a great question. So there is that zone where what I consider in my mind a gray zone. Is that really, that could be, yes, the answer is it could be. Bear in mind that you really need to have already demonstrated that at least one of these already works, right? That one of these AAVs carrying your gene of interest is, you've got studies that show that it works, and basically if all you're doing is switching out serotypes to see if you get better targeting, I think that would fall within BPN Biologics. As long as it can get done within 2 years.
This was so far the last question in the chat.
Okay. Well, we're right at 1 o'clock, so we did well with our time. I want to thank everyone for taking the time to participate in our webinar. If there's any questions that you think of after our webinar ends today, please contact us at BPN Biologics questions at nih.gov, and we will be getting back to you. We hope to hear from you soon, and please remember that the sooner you can contact us regarding the August 9th application deadline, the better it'll be for us to be able to handle all of the questions that we will be getting. Thanks, everybody, again.
Thank you.
Thank you.